In this lecture set, we're going to talk about carbohydrates, and this is an important macromolecule that is made up of a lot of the foods that we actually eat and found in a lot of the foods we eat. Carbohydrates are generally created through the polymerization of monomers, which means that we have these little groups of monosaccharides, which means one sugar. And we typically have and find these in big long chains within some of the foods that we'll eat. And I'll talk to you about some of those different formats and how they work. But there are different subunits of those monomers, and some of those are shown here. So the general organization of these are C, a number of carbons, and then a water group. That's actually why they got named carbohydrates. So the carbon part is here. And this is the carbon, the carbo, and this is the hydrate. So originally it was thought that there was this carbon group and then this water that was floating around and kind of associated with it when they looked at this initially. But now we know a lot more about the structure, but that was how they were originally named because of this organization. So some of the common ones that we'll talk about: glucose, fructose, ribose, galactose. There's a few other, glyceraldehyde as an example here. We'll talk about these in the different forms and how they organize themselves around each other. When we're looking at these X's here, you'll notice that 6, 5, 6, 3, there are these different numbers that those can be associated with, and we'll talk about those. But a lot of times, sometimes they're called tri, tetra, pent, hex, hept, oct. So these Greek prefixes are used to describe the length of that monosaccharide chain, and I'll give you some examples of that in a moment here. We'll also talk about the difference between the functional groups that we find on them in terms of aldehydes versus ketones. Here is a Fisher diagram. These are just a mechanism for organizing these in space so we can look at them and look at the distinguishing features of them. You'll notice that these all are going to be aldehydes. So this is the aldehyde functional group. That's why we went through those functional groups in earlier discussions, so we can distinguish them and distinguish the different types and functional groups here. So there's a carbon here that's doubly bonded to an oxygen at the end of a chain, which is an aldehyde for each of these. And then in between here at each intersection, there's also a carbon, and that's attached to an alcohol group, and then another carbon on the end and an alcohol group. So this is a glyceraldehyde. Here is another group that has 1, 2, 3, 4 different carbons, threose, and then 1, 2, 3, 4, 5, ribose, 1, 2, 3, 4, 5, and 6. So glucose. So some examples of chains as they're getting longer and longer here. And you'll notice we have the oses as well. So you can also say this is a triose, and we can say tetrose, pentose, hexose. So a more generic version of the names to tell you and describe the number of carbons associated with those different groups. These names were done more historically when we started to figure out, oh, these have 3 carbons or 4 or 5 or 6, before we really knew the structure and the orientation. What else is going to become apparent here pretty soon is that there's a difference if we have the alcohol group coming to the right versus the left, and we'll talk about that. So this orientation of the alcohol groups is actually important in terms of what type of compound it is and what the name is that we're going to give to it. Here's one of the most common sugars that we'll talk about, and this is D-glucose. We'll talk about what that D means, but this is the general name for that compound, and this is what it looks like. This is also referred to as an aldose, and that's because it has the aldehyde functional group on the end. You probably have also heard of fructose, and this is D-fructose. And the difference between an aldose and a ketose, or a glucose and a fructose, is that you'll notice that this is actually a ketone on this carbon right here versus an aldehyde on the glucose molecule. So the difference between those two different types of sugars. So here are some common sugars and their functions. The glucose is the primary source of energy for our cells that comes from, this is a monomer of a polymer of starch or cellulose, and that's an example of glucose here. Ribose is a component of ATP and our DNA and RNA components as well. So it's kind of the backbone of our nucleic acid, which we'll talk about in a few lectures from now. And then fructose, present in high fructose corn syrup, for example. This is a different type of compound that we can convert to a glucose molecule by our cells to generate energy. So that you can see there are a lot of different flavors and versions of these different molecules. And you can see here we have galactose and mannose, and both of these are 1, 2, 3, 4, 5, 6 carbons, right? 1, 2, 3, 4, 5, and 6. The same as glucose. And they're both, and all three of these are aldohexoses, right? Aldohexose. That means that we have the aldehyde, and the hex tells us that there are 6 carbons here. Wait a minute. Why do we have 3 names for the same kind of compound? Well, again, there's that distinguishing feature of we have the alcohol coming to the right, left, right, versus right, left, right, versus left, right. So there are differences in these and based off of which direction these bonds are going to be at. And that may seem counterintuitive right now because can't these just flip around and switch? We'll talk about why that doesn't happen and can't happen and why that is a different structure then. Before we get into that identification of the different types of compounds and based off of where these alcohol groups are coming off, I want you to classify this monosaccharide as an aldose or a ketose and then name it according to the number of carbons on each of the atoms. So how do we do this? And first, we need to determine the functional group, right? We see that carbonyl group is on the end, and that tells us it's an aldose. So that we have that group is an aldose, and then we have to determine the number of carbons. So we have 1, 2, 3, 4, 5, 6 carbons. So that means it would be a hexose. So this would be an aldohexose. In the lab, we can actually use different reagents to determine if a sugar is an aldehyde or a ketone, and this is typically done with either Tollen's agent or Benedict's reagent, and these will both, one has silver in it, one has copper in it, and if the chemical reaction occurs, then we know that the component is going to be an aldehyde, for example. So here's an example of this solution. All 3 of these had different carbohydrates in it. This one turned out to be an aldehyde, so we knew that this was an aldehyde of some sort. These examples, these 3, if we have a precipitation reaction, that means that the aldehyde reacted, and so these 2 reacted and this one did not. That would suggest these 3 are different ketones, and these 3 are going to be the aldehyde. So there are different reagents that you can use to identify if something is an aldehyde or ketone in solution, and just kind of an interesting way to do this. This is kind of fun also. Typically, sometimes in organic chemistry lab, we'll take some of those clear glass Christmas ornaments, and then you can put a sugar in that compound that's an aldehyde with the Tollen's reagent, and you can create that mirroring of your glass. So it's kind of a neat way to make Christmas ornaments. Okay, so what's going on with these different compounds and how are we giving them different names just based off of where these different alcohol groups are coming off on this chain? So this is an example of, these are flattened out so that we can see easily the differences between these 2 and how they're oriented in space. But remember these carbons, the aldehyde, obviously this is a trigonal planar, so this doesn't matter in terms of its orientation. But down here in this carbon, what you'll notice is it's attached to 4 different and unique things. So we have this carbon's attached to a carbonyl carbon, an alcohol, a hydrogen, and another carbon that's not an aldehyde. So it's got 4 different groups that it's attached to, which means it's a chiral center. So I'll spell that out for you. And what is interesting about this is that if we're going to try to grab onto this, so if you're an enzyme, for example, you have a certain shape and a certain structure, and it's kind of this idea of if you're a right-handed enzyme or a left-handed enzyme, you'll be able to interact with one of these 2 molecules, but not both. So here's just a quick little image of that. And these are mirror images. You'll notice also that if I kind of draw a line down the center of these, you can see that that would be the same. They kind of are mirror images of each other. Obviously, this would probably be better if that was an oxygen and a hydrogen here, but that's kind of the only difference. It doesn't really matter in terms of that part of it, however. So bear with me for a second. So now let's look at this example over here on the right side. I just want to demonstrate this idea. So if you have your left hand and your right hand, and you're going to kind of cradle this in here, you'll notice that one of the molecules is going to be this COOH group. This would be a carboxylic acid. So this carbon is an analogous compound, right? It's not the same exact one, but we just have 4 different unique things. And we have, if we're going to grab this with our left hand, you can see that that carboxylic acid is going to go in between your index, or this ring finger and your pinky finger. Your thumb's going to grab the hydrogen, and down kind of on your palm is going to be the amine group, and the R group is going to be sticking out straight at you. Now, if you have the other compound, this mirror image compound, this would be able to kind of touch those same points on your hand. But if you were going to move your left hand over and grab this compound, if you need to grab this with your pinky and ring fingers, that COOH group isn't going to be accessible anymore, right? Your hand is going to be, this is going to be more touching your thumb. So it's not going to be able to, maybe you don't have the right information here or the right intermolecular forces to interact with that, and you won't be able to recognize it. So these are actually distinct and different and unique things based off of that tetrahedral shape and how it's oriented in space. If I have an enzyme that can interact with this molecule, that's not necessarily and probably not going to recognize this molecule. So it's an important feature. And what has to happen, what we have to have, are 4 different unique things attached. And on these sugar molecules, all the carbons in the middle are going to be that. Once we get to this carbon down here, we have 2 hydrogens attached. So this is not a chiral carbon. This carbon only has 3 things attached. It's not a chiral carbon. It's not a chiral center. This carbon has 4 things attached to it that are different and unique, so it makes this a chiral center. All right, let's look a little more closely at this and what this means for us now. So if we have this asymmetric, it's the penultimate carbon. That means the second to last carbon in this ring, as we have, usually we're putting the functional group as far up the chain as we can. And then this is the second to last carbon. So this is the last carbon, which is typically not a chiral center because it's going to be a CH2OH group. So this is the penultimate, second to last carbon. And we're going to use that for giving it a name and distinguishing between the different ones. So this, because it's on the left side, we give it the L configuration. And if it's on the right side, the hydroxide's on the right side, it's given a D conformation. So we have the L conformation and the D. This is D instead of an R because it means dexterous, which is a Latin form or saying right-handed. So that's why it's D instead of R. So apologies for that, but that I didn't create the naming system. So L and D configuration. Remember, these are mirror images, and this on the left is a glyceraldehyde, and it's L-glyceraldehyde because of where that hydroxide is. And this one on the right is D-glyceraldehyde because it's on that right side. So this is how we distinguish these different sugars based off of that penultimate carbon where those hydroxide groups are coming off. Glyceraldehyde is one of the smallest and most simple sugars, but you'll notice that there are multiple asymmetric centers in something like a hexose or glucose, for example. This is D-glucose. We're looking at the penultimate, second to last carbon, and that hydroxide is on the right side. It has the D-glucose format there. You'll notice that we have asymmetric sugars at all of these locations in the middle. Obviously, the aldehyde is not an asymmetric carbon because we only have 3 things attached. This carbon has 4 things, but 2 of them are the same, so it's also not an asymmetric carbon. Here we have mirror images of these two compounds, and again, I apologize, this is drawn out so that it's not really given a mirror image, at least on the functional group. That group doesn't matter in terms of the mirror image state, but it's more we're focusing on these different compounds, the OHs, the alcohol groups. So you'll notice that penultimate carbon, that's second to last carbon, we have the D configuration for or the dextrose right hand for D-glucose, and this is L-glucose over shown here. So the difference between D-glucose and L-glucose is pretty important. We can metabolize and consume and recognize this in our cells. L-glucose is not something that we would eat or be able to use for energy purposes, because this small little change here, our enzymes won't recognize this molecule effectively. There are other types of sugar molecules and different types of relationships, and this one is the diastereomer, which means that they're not mirror images of each other. We can't put this mirror in between, and we can see that right at this second carbon here, the O and the hydroxide and hydrogen wouldn't be mirror images. So, but this is a hexose, so it's a similar type of molecule, but we have differences in how these are organized. And we can kind of see here, if we have the same configuration in these carbons, but then mirror images in between. So that's the difference between an enantiomer, which is shown on the previous slide, and a diastereomer shown here. In terms of the Fisher projections and what they actually are trying to represent, you can kind of see how this is organized here. It's a little bit different than what we have been seeing, where that kind of cross in between, that there's a carbon there. And that carbon is supposed to kind of be back into the page or into the screen. And these groups on the sides are actually coming at you, towards you, kind of as shown with these little bars here coming out at you. And then this would be kind of coming back from the page out in towards you a little bit more. So you can kind of see that here as well, where this would be kind of either coming back, or it's kind of this is the shape, this is what's happening. We're kind of lining these up, and depending on where that's going, it's either going into the page or coming out of the page. But either way, this is kind of the orientation and organization that we're trying to represent through this Fisher projection. Rules for looking at this and how to organize this in terms of the drawing these out and determining D versus L conformation is the most oxidized carbon is always placed on the top, which would be the aldehyde or the ketone. And then if the bottom second to last is on the right, it's a D. If it's the bottom second to left on the left, then it's an L. So just kind of reiterating that point there. So we have had a discussion about some of the chemical reactions that can occur between these different types of compounds. So if we have an aldehyde and an alcohol, these can react and interact in forming a hemiacetyl group. And this is an important structure that's going to be seen, and something that's going to be seen a lot in our carbohydrates. What we can do is, if we kind of reorganize the shape of this, so this aldehyde is going to be here, that's the 1 carbon, and then 2, 3, 4, 5, and 6. We're going to reorient this in that kind of more cyclic structure. And you can see how some of these groups go down to the bottom or come up to the top. So these alcohol functional groups are going to be pointed in different directions depending on if they're right or left on this orientation here as well. Once we rotate this and organize this to create this ability for that oxygen to interact with that carbon center, then we can form these different forms of glucose. So we have beta glucose and alpha glucose, and it just depends on how this aldehyde, if it flips down or flips up on that ring structure. So this is a formation of a hemiacetyl group. This is the OH group, and that's the OR group. So this is typically a hemiacetyl group is not very stable. But since this is happening on a molecule that has all of the functional groups all in one, these things are oriented in such a way that this will actually be pretty stable and happen quite often. So typically we're going to be in one of these conformations in a cyclic form because it's very stable in this particular type of structure, because these the reactants are kind of tethered together and help that to be more stabilized. So there are structural differences, and they seem like they would be maybe small, but they have enormous biological consequences. Typically, when glucose is in a crystal form, like sugar, that might be in table sugar, for example, then we would have almost entirely it would be in the cyclic form, and usually even in the alpha form, just for how it organizes itself. In solution, however, it's more often in the beta form, beta D-glucose form, in this orientation. So it spends a majority of its time in either of these conformations, and depending on if we're talking about the solid versus aqueous form, those are going to be dictated by these orientations. And hardly any of it is in that open chain conformation. So glucose is really important for our metabolic needs, and we'll talk about that later in a later chapter. Galactose here is widely distributed in plants and pectins in terms of plant use. It's a disaccharide in lactose, so sugars within milk. And this is also a aldohexose, and it differs from glucose in that spatial orientation of the hydroxide groups. Here is the closed chain form, and here's the open chain, and then the beta form versus alpha and beta, which is this on the number 1 carbon, depending on if that flips up or down on that ring. We also have fructose. I'll show you this is a 5 carbon or 5 membered ring. One of those members is oxygen. Here's alpha D fructose, and again, this is a lot of fruit sugars and honeys, and this is the sugar. These can be converted into glucose in our body so that we can utilize this to generate energy. Open chain form, and then the beta form, where that hydroxide flips up versus down here. So the difference between fructose and glucose, you'll notice that there's an extra carbon hydroxide ring off the end, and that's because that is a ketone that's generating this acetaldehyde functional group. So here you'll notice that is a ketone instead of an aldehyde like the other example. So we have this extra on this carbon, right? This is the 1 carbon hanging off the chain versus being within that chain, within that cyclohexane group. Ribose is another example of a sugar. Here is beta D-ribose, and then beta D2 deoxyribose. And you'll notice the difference here on that second carbon. We have lost that hydroxide group. So deoxyribose nucleic acid is DNA. That's really the kind of sugar backbone that makes up our DNA molecules. So something I'd like you to think about and do kind of so you can work through this a little bit in your head. If based off of the conversion of fructose, so based off of this conformation and orientation, I want you to write out the open chain conformation, so the analogous conformation to D-fructose shown here for the D2 deoxyribose compound. So rewrite that out and post that in our Blackboard space. Now I want to take some of these groups and I want to these are monomers. Remember, they are big long chains of these. So how do we get these to kind of form together and group together into longer chains? One of the way that we do this is through this glycosidic bond, and they can be in the orientation of the alpha or beta form. So here is an alpha D-glucose molecule in the ring form, and here's a beta D-glucose here in the ring form as well. We're going to start looking at how we can join these together, and here are some examples shown over here on the right. And this is when both of these groups are going to form down, and this is the alpha glycosidic bond. So when this group is coming down off of this first group. So this is the 1 and a 4. This is a 1,4 alpha glycosidic bond here. That's going to give us this new group that's called maltose. Here is a beta glycosidic bond, and it's coming up from the 1 carbon, and it's coming down from the 4 carbon. So this is a 1,4 beta glycosidic bond. Remember, we count our carbons from this furthest right carbon group. So it's 1, 2, 3, 4, 5, and 6. And this one would be 1, 2, 3, 4, 5, 6. So this is the 1, and this would be the 1, 2, 3, 4. So that's how I'm getting the 1,4 glycosidic bond. Beta, because it's coming up versus down here on that first carbon. This group here is going to be an alpha glycosidic bond. It would be a 1. And then we start with this carbon for these types of fructose bonds. 1, 2, 3, 4, 5. So it's a 1,5 alpha glycosidic bond. That gives us sucrose. This is actually what is made in table sugar. That's typically what those groups are made up of. These things can be really long. So green plants will often store some of this energy as starches. So they'll store these are kind of a cartoon of these long ring structures glued together. And amylopectin is a type of starch. It has alpha 1,6 branches, and they're really long chains that somewhat infrequently will branch off to different groups. Glycogen stores, this is how we store in our muscles, and most other animals store energy in terms of this glucose within our bodies through these glycogen granules. So these packs of these molecules in glycogen stores. They branch a little more frequently, and you can see we have alpha 1,4 and alpha 1,6 branches that are kind of shown here in this other side picture as well. Our bodies can recognize both of these forms of sugar and how they're organized and oriented. So we can recognize alpha 1,4, alpha 1,6 branches. So we can eat a plant and we can break down the sugar and utilize that. There's another type of sugar or carbohydrate in plants that we actually cannot digest and metabolize in our bodies, and that is cellulose. So this is part of fiber. This is what we want to eat for our healthy, to lower cholesterol and to reduce some of that those issues that we have with our nutrition. And the reasoning for that is these cannot be digested, so they help to get rid of some of the cholesterol, some of our fats through our bile acids and bile salts as they mix and work through our digestive system. So these are not digestible because we have this special link here, this beta 1,4 linkage, which our bodies, we don't have enzymes to break this down. We can't break this down. Now, there are some organisms and things that can digest this. So, for example, cows are able to break some of this stuff down and utilize this as energy. But it takes some time. It takes maybe some other help systems like microorganisms or bacteria that can help break these things down. But most mammals cannot digest this on their own. Other examples of polysaccharides found in different life forms. A lot of bacteria will have different sorts of organizations of these different glycnac and mirinac polysaccharides associated with them in terms of these different types of orientations of beta 1,4, beta 1,3 linkages. And we also have in fungi, we'll also have different types of organizations for different structural polysaccharides as well. We also have chitin, which is a structural polysaccharide that's actually modified on the end with a amide functional group that is coming off of this second carbon here, also using a beta 1,4 bonds. This is called acetylglucosamine, which is the monomer that's used here to create this hard shell of these different types of animals. So these are all different types of sugar groups, and you can kind of see this outlines a bunch of them. Beta-glucosamine, acetylglucosamine. So some of these different functions, there are different changes that are made, right? Instead of just hydroxide or alcohol functional groups coming off, we might have some different side chains that are modified here. So we have some polysaccharide subunits, bacterial wall cell units, mirinac, glycnac, I mentioned earlier, and here are some for chitin, how that is oriented as well. So these are just some different uses of these different types of compounds. So some of these things are found on the outsides of our cells, and they'll be attached to the lipid membrane of our cell surface. So for a cell, whether it's prokaryote, so whether it's a bacteria or it's a eukaryote or multicellular being, what could the value of having this outer layer of carbohydrate be? What kind of properties do you think it has, and what is the value of that? So post this in your Blackboard space. Just kind of write a little bit about what you think the value of that might be in terms of having that outer layer of carbohydrate. Another mechanism that we use as humans for kind of protecting our bodies from foreign substances is that we have cell carbohydrate systems that we organize and generate and put onto our cell surface. So human blood can be classified by some of the oligosaccharides that are placed on the outside of our red blood cells. So you'll notice that if you're O or A or B, if that's your blood type, you're going to have different organizations of the galactose and glycnac and fructose compounds. So you can kind of see here the kind of green, blue, green, purple, green, blue, green, purple, and yellow. So these are different monomers that are organized in different formats and different orientations. So the O antigen is kind of the most simple, and this is the most common in people are to be O for their blood type. If you're A or B, you're changing some of this orientation and this organization of these and what's attached. So we have the A, and then we have that has a unique different GalNAc subunit versus the B antigen just has an addition of this galactose subunit. And this is really important for these different, the cell surface oligosaccharides and our ability to transfer blood or to either be a donor or a receiver of blood. So typically an O antigen, this can be a donor for anyone because we have this same structure is found in all of these. So antibodies wouldn't recognize the same exact thing. However, if you're O blood type and you were donated A or B, that extra group in there would cause some problems in your immune system and it would reject that blood type. So it would attack that donated blood. So that's not good. So if you're O, like most people in the United States are O for their blood type, you're a really good donor, but you can't receive blood from A or B. There's another factor you might have heard of O positive or A positive, B positive or negative. That's the rhesus factor. It's another protein that's present in and on the cell surface. That just means if it's positive, that means that antigen is there. If it's negative, it's not. It's a protein. It's not, this is a carbohydrate. So this is different. The O, A, and B, that refers to a carbohydrate cell surface, and then the plus or minus is referring to a protein that's present or not. So it's a different factor. So there are variations on that carbohydrate theme that you could notice before. We can also, in addition to adding like an amide functional group, like we're looking at those glycnac protein or glycnac carbohydrates that I showed you in chitin, for example, we can have sulfur containing groups. This is an example of what would be a repeating unit of heparin, which is an anticoagulant, so it prevents blood clotting. And this is important, stabilizes different cell culture conditions for creating, for example, stem cell culture. We need some of these things to help with the growth factors that are involved in maintaining those populations of cells. This is kind of an orientation of what's happening in our cells and how we glue on some of those oligosaccharides or polysaccharides onto the surface of the cell. So this would be the lipid membrane bilayer. So this is what all cells kind of have, this lipid bilayer attached to them, and they'll have proteins associated with those as well. And those proteins will be covalently bonded, these polysaccharides. So this all happens within the cell in terms of, I know we're not going through all of cell biology, but you have an endoplasmic reticulum and the Golgi, and all of these things are processed within those intercellular organelles. Some of the important uses for these in terms of cellulose, for example, it provides a really rigid structure for the plant, and you can see that we have really good hydrogen bonding. There are a lot of locations for hydrogen bonding when these are organized in these flat chains. So this provides that structure and helps to hold these units together and creates that orientation within the plant cell. Of course, these also have a way to store quick energy. In animals, for example, we use glycogen stores. In plants, we'll use that amylopectin. So different orientations and organizations, they look a little different, but they're really used for the same thing. It's just if you're a plant or an animal, they're different side chains based off of where they connect to each other. So that's what I wanted to talk to you about in terms of carbohydrates and their structure, orientation, all the things you might want to or need to know, and even some things you didn't want to know about carbohydrates. But the next part, we're going to look at fats and lipids and some of that information about the cell membrane. And we'll continue to kind of connect these issues and start to build up what some of the components are that are part of our nutrition and our diet, and some of those ideas that allow us to recognize how these things are used for energy in the last section of this course.